And of course, you can use DNA to bring two different proteins or two different polypeptides or two different domains together. So, and you can have other examples of this as well. For example, you could encase three different proteins of the same pathway, or you can actually mimic an organelle. Then, of course, DNA protein nanohybrid structures. So you can bring the two together, and you can use the goodness of both the DNA and the protein to your advantage. So the co-assembly of nucleic acids and proteins into functional hybrid nanostructures is a rapidly developing field that utilizes the advantages of both biomaterials while removing their individual disadvantages. So with DNA, you can have the power of prediction. With protein, you can have a lot more functionality. So what we do is we bring them together, and then what we make is structures like DNA cage or DNA vault or DNA tubes or rings with protein core and DNA RNA scaffolds for tethering proteins together in proximity have been fabricated for a range of biotechnology applications. Attachment methods employed to produce hybrid structures are broadly utilized, either covalent chemical conjugation or DNA or RNA binding protein domains. What you can also have is functionalized DNA locks, so that the function is available only when the cage is open. When the case is closed, you do not have the function available. So functionalized DNA locks have been created that are capable of opening or closing in response to environmental stimuli. These stimuli could be pH, light, or temperature, or it could be also in the presence of a specific target molecule like aptamers. We'll talk of that. DNA locks comprise of two complementary single-stranded DNA strands that, while annealed, hold a DNA nanostructure in close conformation. So you have basically DNA locks which are comprised of two single-stranded DNA strands which are annealed to each other, and as long as they remain annealed to each other, you will have a close conformation. But then molecule-specific mechanisms to trigger unlocking include strand displacement and aptamer binding. So strand displacement is basically mediated by three different strands. One of them is slightly longer. If you see, the red one is longer, and this red one is complementary to what is known as the original strand or the protected strand, the blue one. So this is when the blue and red are intertwined with each other, they form what is known as the lock. So then your DNA case is closed. It is closed conformation. However, this red strand also has an extension that is known as toehold, and toehold is complementary to a third DNA strand that is known as the invader DNA strand. And when the invader is present, the invader will start binding or hybridizing at the toehold region, and slowly it will replace the blue strand here, right? So this now is your open conformation here, where the invader strand is bound to the red strand or the toehold. That is your open conformation. You see, the red portion is free again, so this can again bind here, and when the blue strand now starts binding and replaces the gray one, the case can be locked again. So this is like a lock and key where you can open and close depending on which strand, whether the original strand or the invader strand is bound to the toehold strand, right? So TMSD or Toehold Mediated Stand Displacement starts with a double-stranded DNA complex composed of the original strand and the protected strand. The original strand has an overhanging region, the so-called toehold. So the original strand or the red strand is slightly longer. It has an overhanging region. This is called the toehold. Toehold can bind the invader strand, and once the invader strand starts binding and hybridizing, it can progressively replace the blue strand, resulting in open conformation, right? So the invading strand is a sequence of single-stranded DNA which is complementary to the original strand. The toehold region initiates the process of Toehold mediated strand displacement by allowing the complementary invading strand to hybridize with the original strand, creating a DNA complex composed of three DNA strands. So at the intermediate stage, when it is neither closed nor open, you'll have three DNA strands. The toehold has hybridized, but it has still not replaced the blue strand, and over time it will replace the blue strand, resulting in opening. The toehold region initiates the process of TMSD by allowing the complementary invading strand to hybridize with the original strand, creating a DNA complex composed of three strands of DNA. After the binding of the invading strand, the initial hybridized strand or protected strand is progressively displaced. TMSD is commonly used as an on-off switch in DNA protein nanohybrid structures. So here is a DNA sheath, and this DNA sheath is locked by two DNA strands here. So if you see here, this is your double-stranded DNA, and this is locked conformation. This can be opened if you have a invader molecule. The yellow strand as shown here. The yellow strand progressively binds here and replaces the blue strand. And now this opens up. When this opens up, there is a protein inside. This could be any protein. In this case, you have bovine alpha chymotrypsin, which is a peptidase or protein digesting enzyme. This can float in the blood, and then anywhere you want to open it, it can be opened by this single-stranded DNA key, and then the enzyme can perform its action here. Encapsulation and controlled activity of enzyme in hybrid nanostructures. So this is typically a DNA nanavault or DNA nanocase, where a protein is sequestered inside and kept inactive, unless, of course, the DNA case opens. And once it opens, the protein can, if the substrate are present, the protein can convert it into the products. The sequestered enzyme bovine alpha chymotrypsin is inaccessible to its substrate in the closed conformation. The double-stranded DNA locks that hold the vault closed are displaced through competitive interaction with the complementary key single-stranded DNA. So this basically is via Toehold mediated stand displacement that the lock would open. The opening of the vault enables the access of enzyme and conversion of the substrate to the product. So this is one example. Then, of course, what you can have is what is known as aptamers. Aptamers are nucleic acids or proteins. So you could have a question, aptamer is a nucleic acid or a protein. It could be both. Nucleic acid aptamers are highly selective and bind with target with very high specificity. The aptamers bind to its cognate target through various non-covalent interactions, usually electrostatic interactions, hydrophobic interactions, and induced fitting. These are useful in biotechnological and therapeutic applications because molecular recognition properties are just as specific as antibodies. So instead of an antibody, you can use an aptamer, and when the aptamer binding takes place, you can have the release of your drug. So there has to be a biomarker that the aptamer will identify. So you can do a targeted delivery of the drug using an aptamer, right? It can be engineered, and the important property of aptamers is they could be synthetic molecules. They need not be natural molecules. They can be engineered and synthesized by chemical synthesis. They elicit little or no immunogenic response in therapeutic conditions, and they can be used as on-off switch. So let's say you want to specifically target a subtype of cell within a population of large population of cells, a very specific subtype of cells. So what you can do is you can add two different aptamers. So you have a green aptamer and you have a yellow aptamer, and you have a cell which has a receptor. So this cell here has only the receptor for the green aptamer. So your nanocase will not open because you require the presence of both green and yellow for the nanocase to open. Then, of course, if you have the yellow key and yellow aptamer, but not the green key, which means the receptor for green aptamer is not there, then again, this does not open. It will only open when both the receptors for both the aptamers are present. So this is an AND logic gate. So AND means only when both conditions are satisfied do you get to move on and get your results. So this is 1 and 1 equals to 1. 0, 1 is 0. 1, 0 is 0, and 0, 0 is also 0, right? So aptamers could either be oligonucleotides or peptides. So they could be either nucleic acids or peptide molecules that bind to a specific target molecule. Aptamer target specificity can be programmed into a DNA nanovehicle, facilitating controlled drug delivery in response to a specific disease marker. So if you have a DNA lock on your delivery vehicle, which one of the strands is actually an aptamer, then of course, unless the aptamer binds to its receptor, it will not open up, and therefore the drug will not be done. So you have a thrombin protein, and this thrombin protein is in the DNA sheet, and this DNA sheet is in locked conformation because of the double-stranded DNA that is bound to it. And one of these double-stranded DNA is an aptamer that can specifically bind to nucleolin. So when it is in contact with nucleolin, this double-stranded DNA lock would open up. The sheet will become flat, or so as to say, not exactly flat. Basically, it opens up, exposing your thrombin, and then thrombin can cause its reaction on the cells, and basically it would be lysis that would happen. So the nanovehicle consists of a DNA origami tube containing the blood clotting protein thrombin. The DNA tube was designed to remain closed in the bloodstream until it encounters nucleolin overexpressed on cell surfaces, on specifically cancer cell surfaces. One strand in the lock is an aptamer for nucleolin. So this One of the strands here is an aptamer for nucleolin. So when nucleolin comes around, it unwinds and binds to nucleolin, resulting in opening of the of your nano vehicle and exposure of thrombin. And then, of course, thrombin takes over and performs the function. It would specifically trigger the lysis of the cells. That's what it would do, right? So these are the references.